Hey there, science fans! Today, we're diving deep into the fascinating world of viruses. These tiny entities are everywhere, from the air we breathe to the soil beneath our feet. They can infect humans, animals, plants, and even bacteria. Viruses are incredibly small, much smaller than bacteria. Now here's the thing about viruses. They're not technically alive. They need a host cell to replicate. Think of a virus like a set of instructions in a protective shell. Once inside, they turn the cell into a virus-making factory. At the heart of every virus is its genetic material, which carries the instructions for making new viruses. This precious cargo needs protection, and that's where the capsid comes in. The capsid is the protein shell that surrounds and protects the viral genome. It's like a suit of armor for the virus. Capsids come in various shapes and sizes, but the two most common types are helical and icosahedral. Helical capsids, as their name suggests, are shaped like a helix or spiral. Think of a spiral staircase, and you'll get the idea. The tobacco mosaic virus, which infects tobacco plants, is a classic example of a virus with a helical capsid. On the other hand, icosahedral capsids are shaped like a 20-sided die. These capsids are very symmetrical, which provides excellent protection for the viral genome. Many viruses that infect humans, such as the common cold virus and the polio virus, have icosahedral capsids. The shape and structure of the capsid play a crucial role in the virus's ability to infect cells. For example, some viruses have spikes or knobs on their capsids that help them attach to specific cells in the host. Now let's talk about the viral genome. This is the heart and soul of the virus, the blueprint that contains all the genetic information needed to build more viruses. Viruses can use either DNA or RNA as their genetic material. RNA is similar to DNA, but it's usually single-stranded. For example, the influenza virus has an RNA genome. This allows it to mutate rapidly, needing a new flu vaccine every year. Some viruses have an extra layer of protection called an envelope. This envelope is a membrane that surrounds the capsid. It's made up of lipids, fats, and proteins, similar to the membrane that surrounds our own cells. Think of the envelope as a disguise. It helps the virus blend in with its surroundings, making it harder for the host's immune system to recognize and attack it. The envelope is often studded with glycoproteins, which are proteins that have sugar molecules attached to them. These glycoproteins act like keys, helping the virus attach to and enter host cells. For example, the influenza virus uses its glycoproteins to bind to specific receptors on cells in the respiratory tract. However, the viral envelope can also be an Achilles heel. Enveloped viruses are generally more fragile than non-enveloped viruses. They are more susceptible to environmental factors like heat, detergents, and changes in pH. This is why washing your hands with soap and water is so effective at preventing the spread of many viral infections. The soap helps to break down the viral envelope, rendering the virus inactive. Section 5. Viral enzymes. The tools of the trade. Viruses are masters of efficiency. They pack light, carrying only the essential tools they need to replicate. Among these tools are viral enzymes. Enzymes are proteins that act as catalysts, speeding up chemical reactions. They're like tiny molecular machines that carry out specific tasks. Viral enzymes play crucial roles in various stages of the viral life cycle, from replicating the viral genome to assembling new viral particles. One important viral enzyme is reverse transcriptase. This enzyme is found in retroviruses like HIV. Reverse transcriptase allows these viruses to convert their RNA genome into DNA, which can then be integrated into the host cell's DNA. Another important viral enzyme is neuraminidase. This enzyme is found in the influenza virus and helps the virus to bud from infected cells, spreading to new cells. By understanding how viral enzymes work, scientists can develop drugs that target these enzymes and inhibit viral replication. These drugs, known as antiviral drugs, can be effective in treating viral infections. Section 6. The viral life cycle, a 5-step process. Let's see how viruses infect a cell and make more viruses. The viral life cycle has 5 steps: attachment, entry, replication, assembly, and release. 1. Attachment. The virus binds to the surface of a host cell. This binding is very specific, like a key fitting into a lock. 2. Entry. The virus gets its genetic material inside the host cell. Some viruses enter through endocytosis. 3. Replication. The viral genome replicates inside the host cell. 4. Assembly. New viral particles are assembled. 5. Release. New viruses escape the host cell to infect others. Section 7. A tale of 3 viruses: Influenza, HIV, and T4. To illustrate the diversity of viral structures and life cycles, let's take a closer look at 3 specific viruses: the influenza virus, HIV, and bacteriophage T4. 1. Influenza virus. This enveloped RNA virus is responsible for causing the flu, a respiratory illness that affects millions of people worldwide each year. The influenza virus has a segmented genome, meaning its RNA is divided into 8 separate pieces. This segmentation allows for a high rate of genetic reassortment, which contributes to the emergence of new flu strains. The influenza virus uses its hemagglutinin, HA, and neuraminidase, NA, glycoproteins to attach to and enter host cells, respectively. 2. HIV. This retrovirus infects cells of the immune system, leading to acquired immunodeficiency syndrome, AIDS. HIV has an RNA genome and uses the enzyme reverse transcriptase to convert its RNA into DNA, which is then integrated into the host cell's DNA. HIV's envelope glycoproteins, GP120 and GP41, play crucial roles in attachment and entry into host cells. 3. Bacteriophage T4. This virus infects bacteria, specifically E. coli. T4 is a non-enveloped DNA virus with a complex icosahedral head and a tail structure. The tail fibers of T4 attach to specific receptors on the surface of E. coli, and the tail contracts, injecting the viral DNA into the bacterium. Section 8. The diversity of viruses. A world of shapes and sizes. Viruses are incredibly diverse in their structures, genomes, and life cycles. They come in a wide range of shapes and sizes, from the simple helical structure of the tobacco mosaic virus to the complex spaceship-like structure of bacteriophage T4. Some viruses, like the influenza virus, have a segmented genome, while others, like HIV, have a single RNA molecule. Some viruses, like the herpes simplex virus, can establish latent infections, where the viral genome remains dormant in the host cell for long periods. This diversity is a testament to the evolutionary success of viruses. They have evolved to infect a wide range of hosts and to exploit a variety of cellular mechanisms for their own replication. Section 9. The importance of understanding viral structure. Understanding the structure and life cycle of viruses is crucial for developing effective antiviral treatments and vaccines. By knowing how a virus infects cells, replicates, and spreads, scientists can develop strategies to block these processes. For example, knowing that the influenza virus uses its hemagglutinin glycoprotein to attach to host cells led to the development of drugs that block this attachment process, preventing infection. Similarly, understanding the structure of the HIV envelope glycoproteins was essential for developing drugs that block HIV entry into host cells. Vaccines, which work by stimulating the immune system to recognize and fight off specific viruses, also rely on knowledge of viral structure. Section 10. Antiviral treatments targeting the Achilles heel. Antiviral drugs are specifically designed to target different stages of the viral life cycle. Some antiviral drugs, like acyclovir, which is used to treat herpes simplex virus infections, work by inhibiting viral DNA polymerase, the enzyme responsible for replicating the viral DNA. Other antiviral drugs, like oseltamivir, which is used to treat influenza, target viral neuraminidase, the enzyme that helps the virus bud from infected cells. The development of effective antiviral treatments is an ongoing challenge, as viruses can mutate rapidly, becoming resistant to existing drugs. Furthermore, viruses can evolve to evade the immune system, making it difficult for the body to clear the infection on its own. Section 11. Conclusion: the future of virus research. Viruses are fascinating and complex entities that play a significant role in the world around us. While they can cause disease, they are also essential components of ecosystems, playing roles in nutrient cycling and population control. Understanding the structure and life cycle of viruses is essential for developing effective treatments and vaccines to combat viral infections. As we continue to learn more about viruses, we can develop new strategies to prevent and treat viral diseases, and to harness the power of viruses for beneficial purposes, such as gene therapy and vaccine development. The future of virus research is full of exciting possibilities.